Hi everyone, so today we're going to be looking at karyotypes, chromosome variation, and abnormalities in chromosomes. So we're going to look at the variability of chromosomes in terms of size and number in different organisms, and how we could use karyotypes as a visual representation to help us identify chromosome abnormalities. So on the right hand side of the screen now, you will see what we call a karyotype. And this is what you'll find if you look up, for example, human chromosomes. You might get an image like this, and you can see that they are numbered in pairs. So we have chromosome 1, chromosome 2, and so on, and they're in the homologous pairs like we've looked at before. And then you have the X chromosomes at the end if you're female and XY if you're male. However, this is a very neat organization. For the most part, on the left here is what scientists would look at if they're looking at chromosomes. They take this mix mash of chromosomes that they see under a microscope or some sort of imaging technology, and then they organize it into a very neat karyotype. And karyotypes are very useful, especially in identifying chromosomal abnormalities. So when we are looking at chromosomes and we're trying to develop a visual representation of chromosomes, we will start by dyeing them to create a light and dark bands in the chromosomes. And this is what we call chromosomal banding. So the bands show differences in the sequence of nucleotide bases. For example, A and T will create darker bands, whereas G and C create lighter bands. This banding pattern are very specific and consistent. So for example, chromosome 6 will always have a banding pattern that looks like this, and chromosome 9 will always have a banding pattern that looks like this. So we use karyotypes as a visual representation of a full set of chromosomes in their homologous pairs. And we can identify chromosomes to sort them into a karyotype by using the banding pattern, so the dark and light bands, their centromere position, so this part here, and the length of chromatid, so how long each of those chromosomes are. This is really useful for scientists to identify genetic abnormalities if there's anything abnormal about these three things in a karyotype. So a lot of the karyotypes we've looked at have been human karyotypes, but we can create a karyotype for any eukaryotic organism. So for example, this is a visual representation of a canine chromosomes. This is the karyotype for a yellow cardinal. As you can see, there's a yellow cardinal has many more chromosomes, and they have Z sex chromosomes. This is an iguana. So an iguana actually has 34 chromosomes, 6 pairs of macrochromosomes, so these larger ones, and then 11 pairs of microchromosomes. Here we have a type of plant. As you can see, this plant has 4 sets of chromosomes. So they have 4 copies of each chromosome. So as you just saw with all those karyotypes, we have a lot of chromosomal variation between organisms. And you can also see that in this graph. So we have a firefly that has 8 chromosomes, a chicken has 78, a human has 46. So chromosomes can vary widely between organisms in terms of size of chromosomes and genes, the number of chromosomes, as we can see in this graph, or the number of sets of chromosomes. So humans have 2 sets of chromosomes. As we saw in the previous slide, that plant had 4 sets of chromosomes. And we usually display this as a karyotype. When we talk about ploidy, we're talking about the number of sets of chromosomes. So in humans, our somatic cells are diploid, so that's how we would describe human chromosomes as diploid. This means we have 2 sets of chromosomes. We write diploid as 2n, so n means the number of chromosomes and 2 is the number of sets. So we have 2 sets of 23 chromosomes, which is 46 chromosomes in total. Other organisms may naturally have different numbers of sets of chromosomes, such as Atlantic salmon, which are triploid, so we'd write that as 3N. If something is tetraploid, it means it has 4 sets of chromosomes, we'd write that as 4N. In humans, you can have chromosomal abnormalities that lead to polyploidy, meaning there are more than the normal amount of sets of chromosomes. However, this is extremely rare and it's fatal to the fetus. So if you have more than 2 sets of chromosomes, the fetus often dies very early on. But you can also use karyotypes to identify chromosomal abnormalities in terms of an incorrect number of total chromosomes, which is because of an addition or an absence of a chromosome. In humans, anything other than our normal diploid number, so anything other than 46, will be an abnormality. So when we have an incorrect number of chromosomes or incorrect total number of chromosomes, we call this aneuploidy. So this can be an addition or a loss of a chromosome. If we have one missing chromosome, so 2N minus 1, we call this monosomy. If we have one extra chromosome, so 2N plus 1, we call this trisomy. If we have 2 extra chromosomes, so 2N plus 2, we call this tetrasomy. Any of these aneuploidy or chromosomal abnormalities can lead to genetic disorders and in some cases is also fatal for the fetus. So these are some examples of different aneuploidies that can exist. You don't need to know the different examples, but you need to be able to identify an abnormality using a karyotype. So I'm just going to use these examples to show you the different karyotypes, but you don't need to remember symptoms or anything like that. So for example, a common monosomy abnormality is Turner syndrome, and this is where all or part of one X chromosome is missing. So as you can see here, there is an absence of an X chromosome. This occurs in only females, so about 1 in every 2,000 female babies born will have this monosomy condition. And here are the symptoms of Turner syndrome if you are interested. The most common aneuploidy that you've probably heard of is Down syndrome, which is a type of trisomy. We can also call this trisomy 21. That means chromosome 21 has an extra chromosome. So there are 47 total chromosomes. This occurs in both males and females. About 1 in every 1,000 babies born will be born with trisomy 21 or Down syndrome. As you can see in this karyotype, very clearly there are 3 chromosomes or 3 chromosome 21s, and you're only meant to have 2. And lastly, tetrasomy. So an example is tetrasomy X. So there are 2 extra chromosome X's, so 48 chromosomes in total. This rarely occurs. We only have about 100 to 150 cases worldwide. Again, here are the symptoms if you're interested. And this karyotype, you can automatically see at the X chromosome, there are 4 copies. We're only meant to have 2 in females. And this is one example of tetrasomy. So I'm going to leave you with this meme as usual. Write down your questions and bring them to class, and I will see you there.